Good evening, everyone. We are starting our new series on genetics. Uh, the chapter includes genomics in obstetrics, abnormalities of chromosome number, abnormalities of chromosome structure, mosaicism, modes of inheritance, cytogenetic analysis, chromosomal microarray analysis, whole genome and whole exome sequencing, fetal DNA in the maternal circulation, etc. So, what is genetics? Genetics is the study of genes, hereditary, and the variation of inherited characteristics. There is one branch of genetics known as medical genetics, which deals with the etiology and pathogenesis of human diseases that are at least partially genetic in origin as well as their prediction and prevention. Then, what is a gene? Gene is a specific sequence of DNA or deoxyribonucleic acid on a single chromosome that codes for a particular protein through a process of translation. A genome is the entirety of all genes that make up an organism, while genomics is the study of how genes function. So, genetics is the study of genes, heredity, and the variation of inherited characteristics, while genomics is the study of how genes function and interact with one another. Chromosomal, Mendelian, and non Mendelian genetic conditions are reviewed in this chapter. Prenatal and pre implantation genetic testing. PGT is prenatal genetic testing, and PIGT is pre implantation genetic testing, and newborn genetic screening. So, there are three terminologies: PGT, PIGT, and newborn genetic screening. Genetic disease is common. 2 to 3% of newborns have a recognized structural abnormalities, while another 3% are diagnosed with an abnormality by the age of 5 years, and another 8 to 10% are found to have a functional or a developmental abnormality before reaching adulthood. So, if we divide the patients, then 2 to 3% newborns have structural abnormality. Another 3%, in another 3%, an abnormality can be diagnosed by the age of 5 years, and in 8 to 10%, functional and developmental abnormality before reaching adulthood is found. So now, Human Genome Project. This project was an international research program that sequenced around 3 billion base pairs. So this is the MCQ. It has sequenced 3 billion base pairs. And how many genes? More than 20,000 genes have been studied in this project that make up the human genome. It paved the way for research into gene organization and function in an effort to understand the molecular basis of a disease. More than 99% of our DNA is identical. So, among Homo sapiens, 99% of the DNA is identical and only 1% is non identical. There are two regions in a gene. First is coding region and another is non-coding region. The coding region is known as intron, while the coding region is known as exome, while introns are the DNA sequences that are involved in coding regulation. So the coding regions of DNA exons constitutes only 1.5% of the genome. And an exome is the entirety of all the exons that an organism contain. Introns are DNA sequences involved in coding regulation and make up 24% of the genome. Intergenic DNA composes the remainder. So, if we divide DNA into three parts, then we have exons, which is around 1.5%, introns around 24%, and rest 75% is intergenic DNA. Our genetic code varies once every 200 to 500 base pairs, usually as SNPs. SNPs are single nucleotide polymorphisms. Thus, whole genome sequencing and whole exome sequencing holds tremendous potential to further elucidate genetic variants in a disease. Towards this goal, genetic and genomic databases are maintained by the National Center for Biotechnology Information, also known as NCBI. These are freely accessible and can be indispensable to providers who offer genetic counseling and testing for genetic conditions. The Gene Reviews database has in depth clinical information of more than 800 genetic conditions, including their diagnostic criteria and management. The Genetic Testing Registry, or the GTR, contains information of around 80,000 genetic tests. So, Gene Review database has information about 800 genetic conditions, while Genetic Testing Registry, or GTR, contains nearly 80,000 genetic tests and instructions for collecting specimens and transport to individual laboratories throughout the world, according to NCBI 2021. The NLM, or the National Library of Medicine, has also established a genetic information database known as the Medline Plus Genetics. So, three databases are mentioned here: Gene Review, GTR, Genetic Testing Registry Database, and Medline Plus Genetics, which is intended for the lay population. It provides explanation of more than 1300 genetic conditions, a glossary of genetic concepts, and links to more comprehensive medical resources. Now, coming to chromosomal abnormalities. Chromosomal abnormalities figure prominently in the genetic disease. They are around more than 50% of first trimester miscarriages. So, in first trimester miscarriages, the most common cause of first trimester abortion is chromosomal abnormalities, and they account almost 50% of the cases, while 20% cases of second trimester loss So, chromosomal abnormalities, more than 50% chromosomal for first trimester abortions. Most common cause is chromosomal abnormalities, which accounts for about 50% of first trimester losses, 20% of second trimester losses, and 6 to 8% of stillbirths and early childhood deaths are due to chromosomal abnormalities. Based on data from population-based registries, chromosomal abnormalities are identified in 0.4% of recognized pregnancies. Most of these are aneuploidy. Trisomy 21, or Down syndrome, accounts for more than 50%, trisomy 18, or Edwards syndrome, for nearly 15%, and trisomy 13 for 5% of the cases. So, among all the aneuploidies, Down syndrome accounts for more than 50%. So, this is for Down syndrome. Trisomy 18 accounts for around 15%, and trisomy 13 for 5% of the cases. If no karyotypic abnormality is identified in a fetus with a structural anomaly, additional testing with chromosomal microarray analysis is anticipated to detect chromosomal deletion or duplication. This is a CNV, or a copy number variant, which is found in approximately 6.5% of the cases. So, among chromosomal abnormalities, most commonly they are aneuploidies. And which aneuploidy? Downs, Edward, or Patau syndrome. Down is around 50%, trisomy 18% for nearly 15%, and trisomy 13 for 5% of the cases. While if in the presence of structural abnormalities, chromosomal abnormalities is not identified, then we can do one another test known as CMA, or chromosomal microarray analysis, which detects CNV. CNVs are copy number variants, and they are approximately 6.5%. What is copy number variants? Here, chromosome number is normal, but there is part deletion or duplication. Standard nomenclature. Chromosomal abnormalities are described by the International System of Human cytogenomic nomenclature, abnormalities fall into two broad categories: abnormal chromosome number, such as trisomy, or abnormal chromosome structure, such as DNA segment deletion or duplication. Each chromosome has a short arm, which is denoted by P, and a long arm, which is denoted by Q. Short arm is also known as petit arm or a P, and a long arm known as Q. Selected because it is next letter in the alphabet. The two arms are separated by a centromere. When reporting a karyotype, the total number of chromosomes is listed first, chromosome corresponding to the number of centromeres. This is followed by the sex chromosome XX or XY, and then by a description of any structural variation. Specific chromosomal abnormalities are indicated by standard abbreviations such as DEL for deletion, INV for inversion, and T for translocation. The affected regions or the bands of arms are then reported so that the reader will know the exact location. So, look at this table. This is here we have written first the chromosome number, that is 46, and then sex chromosomes, whether the person is male or a female, so XX. So this is a normal female chromosomal constitution. If we write 47, means the total chromosome number is 47, genotype is male, plus 21. Plus 21 means there is one additional chromosome number 21, that is, this person is a male with trisomy 21. Another one, 47XX plus 21, 46XX, means a female with Down syndrome, or a female who is mosaic of trisomy 21 cells and cells with normal constitution, means in this female both normal and abnormal cells are present. So this is a mosaic female. 46XY deleted for P14. This means 46XY means this person is a male with deletion of chromosome number 4 at band P14. P is petit, short arm of chromosome number 4 is deleted at 14 band. So male with terminal deletion of short arm of chromosome number 4 at band P14. This is female with duplication of chromosome number 5 with short arm of chromosome number 14 and 15 with band 15.3. Female with duplication of short arm of chromosome number 5 from band P14 to band P15.3. This is also deletion. Male with balanced male. This is a male. DER 1314 Q10 Q10. So here male with balanced Robertsonian translocation denoted as DER. So Robertsonian translocation is denoted as DER. And which arm is this? Q. This is long arm of chromosome number 13 and 14. So there is Robertsonian translocation between chromosome number 13 and 14 at loci Q10 and Q10. The karyotype now has normal 13, normal 14, and the translocated chromosome, reducing the chromosome number 46 chromosome complement to 45. This is 45XY. 46XX. 46XX with T1122, Q23, Q11.2. So this is a female with translocation between chromosome number 11 and 22 with breakpoints at 11 Q23 and 22 Q11.2. This is an inversion male with inversion of chromosome number 3 that extends from P21 to Q13, a pericentric inversion because it includes a centromere. So this is a pericentric inversion which involves a centromere. 46X RX P22.1Q27. So this female has only one normal X chromosome and one R chromosome. R chromosome is ring X chromosome with the regions distal to P22.1 and Q27 deleted from the ring. R is ring chromosome. 46XIXQ10. Female with one normal X chromosome and one isochromosome. I for isochromosome, R for ring chromosome. Of long arm of another X, ISH22Q11.2 HI array into 2. This is ISH is for fish. Fish of metaphase cells using a probe for the HERA locus of 22Q11.2 region with the two signals identified. There is no evidence of microdeletion. ISH deletion 22Q. There is fish of metaphase cells using a probe of HERA locus of 22Q11.2 region with one single identified consistent with, this is HRA minus, HIRA minus means microdeletion. ARR is microarray analysis. So this is fish analysis, this is microarray analysis. Genome build GRCH38 showing a single copy gain of chromosome 18 from band 11.3 to Q23 to band Q23 entire chromosome consistent with trisomy 18. So this defect is found in 18th chromosome. This is Edwards syndrome. This is microarray analysis of GRCH38 showing a copy loss of long arm of chromosome number 4 through Q35.1, and this is for 15Q. So this is SNP microarray analysis genome builds GRCH38 showing homozygosity for the entire arm of chromosome number 15. So this table is an example of karyotype designations using 2020 International System of Human Cytogenomic Nomenclature. Nomenclature is similar to fish. Fish is fluorescent in situ hybridization, a technique which is rapidly used to identify chromosomal abnormalities. ISH is used for fish or in situ hybridization performed on the metaphase cells, and NUC-ISH is performed for interphase nuclei. So if plain ISH is used, this is for metaphase fish, and if NUC-ISH is used, then this is used for interphase nuclei. If the result is normal, ISH is followed by a probe's specific chromosomal region such as 22Q11.2 and then the name of the probe and the number of signals visualized. For example, HERA 2 times. If the deletion is identified, del is included before the chromosomal region and the name of the probe is followed by a minus sign. If there is deletion, then minus sign is used. The 22Q deletion syndrome is discussed in table 315. For chromosomal microanalysis, so this is for fish. Next is for CMA. CMA is chromosomal microarray and the designation we use for CMA is ARR. This is followed by the version of the genome built to which the nucleotide designations are allied, such as this is GRCH. So we have used this version of genome, which is Genome Reference Consortium Human Build 38. Next, the number of chromosomes containing the abnormalities listed, followed by the P arm or Q arm, and then specific bands in question. CMA reports also include the affected base pair coordinates and thus convey the exact size and location for every abnormalities identified. The information is reported in the same way whether the alteration is pathogenic or of uncertain clinical significance. Now, abnormalities of chromosome number. The most easily recognized abnormalities are numerical abnormalities. So, abnormalities of chromosomal abnormalities are divided into abnormalities of chromosome number and abnormalities of chromosome structure. Coming to abnormalities of chromosome number, also known as aneuploidy. So what is an aneuploidy? Aneuploidy is the inheritance of either an extra chromosome resulting in trisomy or loss of chromosome resulting in monosomy. This differs from polyploidy where whole haploid set is added and which is abnormal number of the whole haploid chromosomal sets. For example, triploidy has 69 chromosomes while trisomy has 47 chromosomes. Monosomy has one deleted chromosome or 45 chromosomes. The estimated incidence of various numerical chromosomal abnormalities are listed. Coming to autosomal trisomies, what is an autosomal trisomy? Where 44 out of 44 out of 44 chromosomes, one chromosome is duplicated from the autosomal genes. Trisomy usually results from non-disjunction when there is abnormal separation during meiotic phase, which is the result of failure of normal chromosomal pairing and separation during meiotic phase. Non-disjunction may occur if the chromosomes fail to pair up properly but unable to separate, or separate prematurely, or fail to separate. So these three conditions can give rise to autosomal trisomies. Although each chromosome pair is equal likely to have a segregation error, trisomies other than 20, 21, 18 and 13 rarely results in term pregnancy. So if there is a trisomy of chromosome number 21, chromosome number 18 and chromosome number 13, then a viable pregnancy can occur. But if other than these three chromosomes there is trisomy of any other chromosome, then it can cause miscarriage or abortion. Each of these autosomal trisomies has a spectrum of phenotypic severity. Some fetus come to attention earlier in a pregnancy due to multiple major organ abnormalities or hydrops fetalis. Others may have subtle findings and are detected only through prenatal screening test. So there is a spectrum of phenotypic severity which can range from hydrops fetalis to subtle findings. Survival data based on cases with better prognosis and other factors includes counseling. The likelihood that a pregnancy will be complicated by an autosomal trisomy rise steeply with maternal age. So maternal age, particularly age more than 35 years, is an independent epidemiological risk factor for chromosomal abnormalities. From birth until ovulation, oocytes are suspended in the mid prophase of meiosis 1. If non-disjunction occurs after completion of meiosis, one gamete will have two copies of the affected chromosome, which can lead to trisomy if fertilized. The other gamete receiving no copy of affected chromosome is nullisomic and can cause monosomy. So if we have two sets of chromosome, one will have one extra pair and one will have deleted pair. So this can cause trisomies as well as monosomies. If fertilized, it is estimated that 10 to 20% of the oocytes are aneuploid secondary to meiotic errors compared to 3 to 4% of sperms. So if we compare the age of a male and a female, then there are 3 to 4% chances of aneuploidy secondary to meiotic errors of sperms, and 10 to 20% in a female with age more than 35 years. So after a pregnancy with autosomal trisomy, the risk for autosomal trisomy in a future pregnancy is approximately 1% until the women's age-related risk exceeds this. Screening and prenatal diagnosis of autosomal trisomies include parental chromosomal studies are not indicated unless the abnormalities was caused by unbalanced translocation or any other structural rearrangement. Looking at this image, figure number 16.2, prevalence of autosomal trisomies according to maternal age is found in a population-based birth defect surveillance programs in United States. So with age, at less than 20 years of age, the prevalence per 1000 live births for Down syndrome is around 10%. Then it is rising with age, and at 35 years, at more than 35 years, the risk is increased to around 30%. Now let's study a little about trisomy 21 or Down syndrome. So what is a Down syndrome? Langdon Down described a group of intellectually disabled children with distinctive physical features nearly 100 years later, demonstrated that Down syndrome stems from an autosomal trisomy. Trisomy 21 causes 95% of the Down syndrome cases. The non-disjunction event occurs during meiosis 1 in approximately 75% of cases. So coming to etiology, non-disjunction occurs in around 75% cases, and this non-disjunction occurs in meiosis 1, and in meiosis 2 in the remainder, and in 25% cases non-disjunction occurs in meiosis 2. Because chromosome number 21 is acrocentric. Acrocentric means its centromere lies at the periphery. So this chromosome number 21 is acrocentric. A Robertsonian translocation may occur. So another etiology is Robertsonian translocation. This accounts for 3 to 4% of Down syndrome births. The remaining 1 to 2% occurs due to I21 or mosaicism, which is isochromosome 21, which constitutes around 1 to 2% of the cases of Down syndrome. So coming to etiology, there are 3 causes: non-disjunction, which can occur in meiosis 1 and meiosis 2 also. More cases are seen in meiosis 1, which is around 75%, and rest 25% are associated with meiosis 2. Robertsonian translocation in 3 to 4% cases, and isochromosome 21 or mosaicism is seen in 1 to 2% of the cases. So this image is showing a male karyotype with trisomy 21. So there is an extra pair of chromosome in the chromosome number 21, 1, 2 and 3, which is consistent with Down syndrome. Down syndrome is the most common non-lethal trisomy. It is identified. So what is the incidence? Incidence is 1 in 450 pregnancies. In United States, fetal loss and pregnancy termination yields an estimated prevalence of 1 case in 740 live births. So in US, the incidence is a little higher. In the world, the prevalence is 1 in 450, while 1 in 740 in US. The fetal death rate beyond 20 weeks of gestation approximates 5%. Coinciding with the rise in overall number of pregnancy in women more than 35 years or older, the proportion of Down syndrome has increased approximately 33%. So due to late marriages and late pregnancies, the incidence has increased around 33% during the past 4 decades. Most Down syndrome births in Parkland Hospital now occur in women in this age group. So if the major anomalies and minor aneuploidy markers are considered, 50 to 75% of the pregnancies are affected by Down syndrome are found to have a sonographic abnormalities. So only 50 to 75% cases can be detected through ultrasound scanning, or sonographical detection is only seen in 75% cases. Rest 25% cases may be missed on a ultrasound scan. A major structural malformation is detected in the second trimester is approximately 1/3 of the cases. The most prevalent anomalies in affected children are cardiac defects and GI defects. So most common or most prevalent findings that we see in Down's baby on a scan are cardiac defects and GI defects. Cardiac defects approximately in 50% of the cases, and GI defects in approximately 40% of the cases. So what are the cardiac defects that are seen in Down's babies? It includes endocardial cushion defects and VSDs. Endocardial cushion defects and VSDs are ventricular septal defects. While GI abnormalities are seen in 6 to 12% of the cases, and it includes atrasias, either esophageal atrasia, duodenal atrasia, or Hirschsprung disease, where there is anal atrasia also, or there is defective migration of neural cells in the intestine. Other health problems are also common in Down syndrome. What other health problems are seen? Hearing loss in 75% of the cases, optical refractive errors in 50% of the cases, cataract in 15%, OSAS or obstructive sleep apnea in 60% of the cases, and thyroid disease in 50% cases. And there is risk of leukemias or transient myeloproliferative disorders in 10% of the cases of newborns. Alzheimer's disease, because the gene responsible for Alzheimer's disease is located in chromosome number 21, and when such babies have extra 21 chromosome, then early onset Alzheimer's disease is also seen in this condition, which is around 70% of the adults and higher incidence of leukemia. Women with Down syndromes are fertile. Nearly one third of their offspring will have a Down syndrome. Males with Down syndrome have markedly reduced spermatogenesis and are almost always sterile. So, if we compare the fertility status between a Down's female and a Down's male, so females are fertile while males are infertile, because in males spermatogenesis is markedly reduced. The IQ score is 35 to 70 in such babies, and social skills in affected children are usually higher than predicted by their IQ scores. Characteristics features of Down syndrome are shown in figure 16-14. So, this is a newborn with a Down syndrome with karyotype 47XX21. A characteristic facial feature can be seen like epicanthal folds, increased palpebral distance, flattened nasal bridge, and you can see the hand. There is just single palmar crease. This palmar crease only present, while in normal babies two palmar creases are seen. So only single palmar crease is seen in this baby, and there is hypoplasia of the middle phalanx of the fifth digit. So this is the middle phalanx and it is hypoplastic. So fifth digit's middle phalanx is also hypoplastic. Now image C, this represents the sandal gap. So this is known as the sandal gap. Sandal gap is the gap between the first and the second toes, also known as hallux varus, which is seen in these babies. Typical findings includes brachycephaly or the small head, epicanthal folds, upslanting of palpebral fissures, Birchfield spots, which are the whitish spots on the periphery of the iris, a flat nasal bridge, and hypotonia. The infants often have loose skin at the nape of the neck, a prominent space or a sandal gap between the first and the second toes, short fingers, single palmar crease, clinodactyly of the fifth digit, in which there is hypoplasia of the middle phalanx causing inward curvature of the fifth finger. So these findings can be seen in prenatal ultrasound examination. Data suggests that approximately 95% of the live-born infants with Down syndrome survive in the first year. The 10-year survival rate is at least 90% overall and 99% if major malformations are absent. Average life expectancy has been increased in the previous decade to around 50 to 60 years. Several organizations offer education and support for prospective parents faced with prenatal diagnosis of Down syndrome. These includes March of Dimes, National Down Syndrome Congress, and National Down Syndrome Society. So these societies offer education and support for such parents having babies of Down syndrome. Next syndrome is trisomy 18, also known as Edwards syndrome.